Uh, really hope you guys had a good lunch and had the Mysore Pak. It was good. While he sets up, Dr. Rajesh Pandey is a faculty at IGIB, Indian Institute of Genomics and Integrative Biology, which is a CSIR institution in Delhi. And Dr. Pandey basically did his PhD at IGIB, and then a postdoc at Hopkins and Oxford, and then came back as a faculty to IGIB. He's interested in pathogen genomics, and during the pandemic, he was one of the first people, like Chitra, got involved in pathogen sequencing. He made they did a very interesting effort where they actually took a van to the Delhi airport, and they were sequencing variants to look at new novel variants which might be coming in through travel into the country. And actually, Rajesh, Chitra and I also have a history. About 3 years ago, we put together a program to do unknown pathogen discovery in the Northeast, and we are still waiting to hear about its fate. So great to have you, Rajesh. Look forward to hearing.
So warm good afternoon. It's not so warm in Bangalore, but then we can say good afternoon. Because I come from Delhi, and Delhi can be defined how summer should be or is in India at the moment. So we feel very good about it at the moment because from 50, 52, we are now 37, 38 in Delhi. So it's very comfortable summer for us at the moment. Yeah, it was. And my wife was doubting whether our home ACs have gone wrong because they were not doing the desired cooling as it was supposed to be. Then we realized that it's not their fault. It's the ambience air which was killing them. So the same ACs have become better in last 1 week. So they are doing their job in terms of cooling. So while I was listening to conversation here, I pulled up a couple of slides just to share a couple of learnings and perspectives wherein as we move along, as we grow older, as we grow wiser, few things go in parallel. One is science, another is economics, and third is partnerships. So partnerships, scientific partnerships, academic partnerships, like Uma mentioned about me, Uma, Chitra, knowing each other during the pandemic for a very specific purpose, we came together and we have carried that partnership. Another is industry partnership where we are almost forced to use reagents from the companies who are selling those reagents. My being a whole and whole genomics lab, we don't do anything else but only genomics. So we work quite closely with many companies which sell these reagents. So many a times, lot of things is a gray area between where a particular technology stands, what another company wants you to believe about another company, and what is the actual scenario vis-a-vis the third company. So in that domain, I thought maybe I will just pull up a couple of slides, which I normally don't show now, because when we were given, as a lab, we were given the responsibility for doing SARS-CoV-2 genomic surveillance by Government of India for whole north of India. And incidentally, it happened, SARS-CoV-2 came at a time, first 2 samples in the lab came on 27th February 2020 to IGIB. And that people who are working in government institutes would know that that's also the end of a financial year. So you also know that your budgets are literally not available for you to buy many things. Your freezers don't have too many reagents because you are at the end of the funding cycle. But SARS-CoV-2 came at that time, and we were supposed to come up with a strategy to do sequencing. And that also the right way. And if we rewind the clock back to February 2020, we didn't had so many protocols or so many optimized protocols. Forget about COVID-sec and other panels which came later on. So what was the flexibility we had at that point of time to do the job which was assigned to us and those 2 samples which came to the lab. So while we did what we had to do at that point of time, reading overnight and putting towards a protocol based on whatever we knew by working in this domain for 2 decades now, that what may work in terms of sequencing. So we did de novo sequencing for those 2 samples, and we did manage to assemble SARS-CoV-2. But at the same time, we also understood that probably this may not work for a country of our size, especially given the population denominator which we are. So we need to come up with alternate strategies, which is more amenable not only in terms of technology, but in terms of making people learn, implement it on the ground, as well as reach to the nooks and corners of the country. So at that point of time, we thought nanopore probably would be a good strategy to work on. So when we started, even in my institute at that point of time, we were told that, oh, nanopore has a lot of error rate. It doesn't work for high CT value samples. And we said, okay, it may be true, but it may not be true, because have we tested for it? So some of the work which we initially did was that not only we set up this pipeline in the lab in the month of end of Feb, first week of March, but going forward, what we also tried to do with initial set of samples, we took different CT value samples and tried to see whether how much of this is dependent on high and low CT value, and how much is potentially related to the RNA quality, or whether it is dependent on the platform being used for sequencing. So what you see here is basically on this graph that these are CT values, and this is the performance of whole genome sequencing of SARS-CoV-2. What you take home lesson message from here is that we find that some of the samples which provided us whole genome coverage were of high CT value, and some which didn't provide were also of low CT value. So we didn't find any significant correlation to say that high CT value sample will not give us a whole genome coverage or whole genome sequencing for SARS-CoV-2. And the same, the reverse is true, that low CT value means high viral content indicator is going to give us a successful sequencing always. These are all nasopharyngeal swabs. So we did this activity in the lab to convince ourselves as well that because you have to rewind the clock almost 4 years and think at that point of time, there was a strong debate that what will be the cutoff which we should take forward for sequencing the samples. So what we did initially to show using 122 samples that this is not a straight correlation. Second point, which was again in the public at that point discussion, was that the quality of reads from nanopore are not of same quality matched for Illumina. So we thought, okay, let's try that out also in the lab. So for initial 122 samples in the lab, we sequenced same 122 samples both on nanopore as well as Illumina, and try to see that, okay, how the mutations are matching with each other, or are there new mutations being called in either of the platform. So what you see here, the green ones are the ones which are matching in both the platforms, and the mismatch are marked by gray. So what we saw during this work, which we never published because this was more for benchmarking the work in the lab and in the institute as well, that it depends on few factors. One, the question that in a pathogen genomics domain, if you're looking for a study like or an investigation like genomic surveillance, you are not always looking for very low frequency mutations. You are looking as an opportunity to identify the mutation, identify the organism, virus, and in those cases, nanopore platforms have perfect matching with Illumina in terms of the mutations if it is more than 1% frequency. If it is below 1%, then of course you capture more in Illumina. But there is second caveat to it. This was sequenced using amplicon-based approach, and this was sequenced using shotgun, de novo sequencing-based approach. So there is also a component of how do you make the library as well. But in terms of identification of the virus, in terms of its lineage assignment, both were 100% complementary with each other. So that was important understanding at that point of time, because we were able to convince ourselves and to the community going forward that what we need is to identify what question we are asking. What was also happening in the background, that in this, around this time frame, Illumina's COVID-sec panel was not there. So what Illumina guys used to tell, slowly around your ears, not in your ears, but around your ears, that nanopore has a more error rate. What they will not say is that you don't have an alternate to nanopore at that point of time. Why? If I do amplicon-based approach on SARS-CoV-2, our lab cost for sequencing a SARS-CoV-2 genome was 3,000 rupees per sample. This is without COVID-sec. If I do using de novo approach, my costing was going into double digit in terms of 1,000. So those were the numbers which company people will not say. They will not go into the details. But as a user, we have to enrich ourselves with those questions, counter questions and queries as well, that, oh, how would you sequence it? Even after COVID-sec came into the market, still per sample SARS-CoV-2 genome sequencing on Illumina is 5,000 rupees compared to nanopore's 3,000 rupees what we do in the lab. Still you have 2,000 rupees gap, and if you're talking about scale, 10,000 sample, 20,000 sample, you are looking at a very high number. So those points which I briefly interjected in, before that as well, that we also need to consider those things while taking a call on this. Listen to the companies, but listen to yourself. And then ask questions to get answers which will suit your requirement. When I say your, it means the mandate of the lab, mandate of the institute, and if it is given by an organization, then the mandate of the organization. Because during SARS-CoV-2 period, we almost sequenced close to 50,000 samples in the lab. If I look at a cost difference of 2,000 rupees per sample across 50,000, it's a huge number which I'm looking at. So thereupon now in the lab now, we use nanopore for pathogen genome sequencing almost without any hesitation for all host response studies, RNA-seq, whole genome. We use high throughput nanopore Illumina platforms like NextSeq or NovaSeq. For example, when we do single cell transcriptomes, of course we use NovaSeq. And now we will come to something what we have now recently invested to move beyond or move in addition to Illumina as well. There are some alternatives available to us. We are now in the process of trying them out in the lab to have more specific applications on those different platforms as well. So this is something which I just wanted to share based on a work which we did in the lab, so that we take suggestions, but of course test it out with your own hypothesis that what works best for me. So with this background now, I will go back to the original slides which I wanted to share today. Assume this, yes. So as a research discipline in the lab, what we do is we try to do something in the area of integrative genomics of host pathogen. And I'm slightly in love with acronyms, so you can excuse me for that. So when we were looking for what would be the mandate of the lab, we thought, and when I was growing up in research as a PhD student, I didn't ever like people starting talk with literally scaring the person on the other side. Cardiovascular, this many people dying. Diabetes, this many people dying. The person sitting on the other side would almost think that I will die tomorrow. So I am very careful that, okay, we all know there is a lot of problems in life. Can research, what we do in the lab, in the institute, in the country, whichever we are, where we are talking, is there some hope we are generating out of that data? That, okay, this understanding probably holds some hope for this. Of course, it will need further refinement, further polishing, further implementation, further strategies. But is there something positive about it? If I just need to reiterate how bad it is, then probably I'm not needed. Anyways, all of us will die one day, that's fine. That's the accepted truth of it. So can the research in the lab provide some hope out of it? So that's why we put it as integrative genomics of host pathogen. We work exclusively with clinical samples coming from hospital partners. And one of the line which we keep talking in the lab, that whatever we do in terms of time spent, in terms of money spent, in terms of intellectual time spent, in terms of PhD students' time, project staff time, are we doing anything which can go back to the patient, go back to the community in some time, not tomorrow possibly, because that also we want to be protecting ourselves, that we don't want to say that, okay, we have found a solution for this tomorrow. No, it's not. If it was so easy, it could have been done by now. I'm not the first person in the world to be doing it. So some of the themes which we do in the lab, which in our opinion is very integrative, is something like this. What we do is like primary work in the lab is around surveillance, genomic surveillance. You can also call it as clinico-genomic surveillance. So we worked a whole body of work during COVID times on SARS-CoV-2. Now we are focusing heavily on dengue, and some initial work on MTB has started in the lab. So we are now only knowledgeable about these three to some extent in the decreasing order. So for us, any genome per se is a genome. Any organism is a genome which will have some size, and we want to understand, is there any information which is encoded in that genome which can be decoded by us through sequencing followed by analytical strategies downstream. So we use pathogen genomes to ask different questions, both in the clinical surveillance and community surveillance domain. And these are different streams which germinate from there, like pathogen genome surveillance, then use that genome information to see whether we can understand transmission patterns out of it, track the evolution of the pathogen, then whether this can be applied in terms of early detection of emerging threats, to understand transmission dynamics of disease across species, and then whether they can be influencing the treatment strategies. And then these are representative work from the lab, which is like, okay, in this domain, this is something which we have done. And these are just representative publications, and I am very bad in that in terms of all these slides which you will find, I have only cited lab work. Because as a researcher who had option to come back from Hopkins and Oxford to settle in India, I think very often we cite work from outside. I think we should respect the work which we do in India. We should also cite each other work from India. And I'm very strongly feel on it, that we don't cite adequately our own work unless somebody else, a paper from Harvard is much better than a paper from NCBS or IGIB. I'm not for it, sorry. Next part which we work is in the area of, okay, a pathogen has infected 100 people. All of them don't give symptoms. Few of them give symptoms. And of those few of them end up having a severe form of the disease or end up in ICU, or even a smaller fraction don't survive. So we try to understand heavily on what's the host factors which may be influencing that, in which again we break up into different hierarchies. And these are independent PhD problems dealt by individual students in the lab, looking at what's the possible host pathogen interaction. So looking at RNA-seq based understanding of gene expression patterns. Then what we also, this is something which is very close to my heart, is like when we initially did RNA-seq for patients, we realized that after our best of effort, 65–70% of the RNA-seq reads only map to human. So don't listen to me, ask me if you have queries. I'm very bad at speaking long. So we thought, okay, maybe sequencing quality was not good, maybe something this, that. So we tried to negate all the reasons, and still only 65–70% of the total reads map back to human. So we thought, what is this? What is this leftover? I mean, we did all the best we could do. So then we started to investigate what's that leftover data. Then we said, okay, we took that leftover data and mapped it to all the available genomes which is there in the database. And we found that a very high percentage, 20% of it almost maps to different bacteria. Around 5% maps to different viruses, and other 5%, depending on different background, maps to some archaea or some other organisms, fungi. So we said, okay, this seems to be an interesting scenario. So we started to invest a little bit more in terms of computational means to understand the biological relevance of it. So a program which we now run in the lab, we call it as transcriptionally active microbes. So if I take a sample of mine and do a transcriptome sequencing of it, the strategy is such that it will sequence anything which is there as RNA. And then you have your bioinformatics toolkit to try to understand what are the subcomponents of it. So the one which is there in the RNA, it means that those bacteria are getting transcribed. So what we are trying to understand is a different variant of metatranscriptome, where we don't do metatranscriptome as an exclusive exercise, but we try to infer it through the host transcriptome itself. And those we have used those data set to explain in SARS-CoV-2 and now in dengue that how those are very significantly associated with the disease severity. And one example which comes to my mind is in one of our initial work, we found that one bacteria, Achromobacter xylus oxidans, was very significantly associated or abundant in the severe patients who died due to SARS-CoV-2. So we then looked for reasons that why it is so, that why this bacteria is present in such high amount in these individuals only. And what we found through literature is that this is known to be highly drug-resistant bacteria. So possibly the hypothesis which we proposed is that all these patients were treated with the same combination of antibiotics, but the effectiveness of the antibiotics on these patients did not have great desired effect because these bacteria did not allow it to happen because they were present in that high amount. Then we did all subsequent studies looking into vaccination breakthroughs and non-vaccinated infections, comparisons across pre-VOC and variants of concern, Delta, Omicron on those lines. Third aspect which we are looking is that what's the role of genetic makeup of the host itself. So looking at genome-wide association studies, that the people who were infected with SARS-CoV-2, if they have different severity, 4 normally we have taken in our studies: mild, moderate, severe, and patients who didn't survive mortality. So we have tried to look into that whether there is a genetic variant in those individuals who didn't survive. So the answer to the question which we are asking, that why a group of individuals infected by same or similar pathogen have dissimilar disease severity. So people who get infected with SARS-CoV-2 or dengue or don't have similar severity, or everybody doesn't end up in hospital. So why it is so? So we feel that there is a combination which is at work going forward. Third aspect which we started 3 years back was looking into single-cell transcriptomics. So during initial studies in the lab, we realized that RNA-seq is a good way to look into the immune profile, but probably not the best way, given the heterogeneity of immune cell types. So we started asking ourselves that, okay, how do we investigate better in that? So we started program of single-cell transcriptomics. So that's a full-blown program now, which we run in the lab. But we have tried to make it a bit more interesting, at least in our own understanding, is in addition to single-cell transcriptome, we also do antibody sequencing, which is like if I'm studying, say, SARS-CoV-2 or dengue, there are some immune markers, surface markers, which are known to express on the immune cells. So we have, there is something called antibody-seq. So there are probes and antibodies which have tags, indexes attached to it. So you can index those surface markers and then pull them and do sequencing. So basically through sequencing, you are able to understand that what is the diversity or the abundance of those surface marker proteins which are essential for immune response in the individuals in which, or the disease in which we are studying. So we coupled single-cell transcriptomics with antibody sequencing, and going forward now we are making it a bit more complex by including TCR and BCR also with that to try to understand what is happening there.
Sir,
Yeah.
go ahead. Sir, how specific this antibody sequencing is? Like suppose I'm doing some clinical trial and I want to differentiate between antibody from external response and vaccine specific response. Can it be different?
Yes. So so this is antibody sequencing is not de novo. So this is like for well-validated surface marker proteins. For example, in response to SARS-CoV-2, we know that these are the predominant surface markers which are expressed. Or in response to dengue, these are the predominant. So in case of SARS-CoV-2, we used 48 panel antibody sequencing to understand the diversity and the abundance of them in the patients which we studied. Now that panel has increased to 60 because there are now more antibodies which has been linked, more proteins which has been linked with their possible functional role in these diseases. So it's a growing process where you have more evidence to know that, well, these proteins are functionally associated with the disease progression, disease pathophysiology, and then whether those are available in the platform which we're using. For example, all of our single cell studies, we are using BD Rhapsody as a platform. Now, whether BD Rhapsody has those in their panel to do sequencing for those antibodies of interest. Going forward, if there is something which is not in the panel, but you want to check it out because based on your work or your hypothesis, you feel that it's not difficult to do. You just need to, if there is a good antibody available for that protein, you just need to attach index to it. You can talk to the companies, they can do it. We have tried to do it for few proteins which we think would be important to investigate. And now since we have been working with them for 3 years, they have been able to work closely with us on that. So we started with 48. Now we have increased that panel to 60. But these are known proteins with known functional role in the disease context which we are talking about. But what is important to note here that you are discovering the role of protein, but not through Western blot or ELISA. You are discovering it through sequencing. So that's an application of genomics which is also worth noting. So, yeah.
The technique that you were speaking, where you try to pull down the proteins associated with the antibody, is it similar to that of affinity capture?
Yes, but at a very nano scale. Okay. Answer is yes,
yes, okay.
But it happens in basically nano wells.
Okay, fine.
Function remains the same.
Yeah.
And it's a very good question because for all of us to remember, whatever we do now in terms of high throughput, the basic technologies is a miniaturization which has happened.
Principle lies the same.
Yes.
Okay.
Your magnets have become much sensitive. So all those ampere beads who are doing experimental people, those ampere beads are what? These are all optimization of sensitive magnetic properties, how you can play around with those different sizes dynamics. But the principle remains the same. Same is case here as well. You are using that affinity or that charge on that with your magnetic strands which are now better sensitive to that to do all those segregation experiments. Yes. So again, we use all those information to understand that, well, can we explain or understand the disease better? And this is a new addition to the lab. Why? Because some of discussions which we had in the lab, and we had some scientific disagreements in terms of how we are approaching AMR as a whole, because I think there is a bit of a rush in how to look for AMR. But we are trying to put up a hypothesis that probably we should look AMR from the transcriptional perspective as a possibly making a case that it may be more functionally relevant. For example, at this moment, if I do my whole genome sequencing, I may have a mutation which may be associated with cancer. But I do not have cancer at this point of time. So there are some balancing things happening. Same way, if there is an AMR allele sitting within my microbiome, is it worth to be taken into account at this point of time unless it is expressing itself as a functional molecule? So we are trying to understand AMR from whether it is getting transcribed from the genomes where it is encoded. So we are trying to look at the AMR from the transcriptional perspective, possibly with a chance that it may be translated into a functional role going forward.
But for that, I think selective pressure is also important, right? I mean, we could look it into the other in other perspective where the gene is present, it is not expressing until and unless we introduce the antibiotic into the system. Only then the functional character
Yes.
comes into the picture.
Yes,
Uh,
you are right.
yeah.
So that's where what I have not said, which I'll talk later, is that in the background of all the samples which we do, there's a whole lot of clinical data. So we are very, very insistent on getting clinical data to the highest detail possible. Of course, we are not generating that data, but we have few very trustworthy, enthusiastic clinical partners who have been happy to collaborate to share very detailed clinical data, which includes the whole battery of drug administration, whole battery of tests being done in terms of readout. And then what we are now trying to go forward, trying to investigate forward is what is the role of diet? So the food which we eat and the amount of AMR which is known to be in the soil, we ultimately eat those food. Now, based on patient's diet profile, how about a possible flux happening between my diet and my AMR profile? So we are now requested the clinician to please capture the dietary profile as well. And dietary profile, if you see, at much valid for India, that if you simply draw it from Middle East, West, North, South, we are so different in terms of dietary profile. What role it may play? So we may be looking at AMR from a very focused bacterial perspective, but probably we should expand our net and look at the other more real-time players, because food is something which without choice we eat 3 times a day. And based on which part of the country we are and what sort of a dietary preference I have, I may not have a choice but to have higher or lower component or quotient of AMR. So that's one thing which we're trying to see, that whether the investigation towards AMR needs a relook or needs a newer perspective. And if you combine what I spoke for last 15-20 minutes into all of them together, then probably we are trying to look into or understand a problem which may help us get closer to the One Health domain. So that's a bit of a prelude with respect to the different research themes which we do in the lab. Now, I will be couple of thoughts which I heard when I was sitting is about genomics being a costly exercise. Yes, it is given that it is a costly exercise, but I'm trying to, I will share a perspective for you to decide, debate, and then practice in future, that how do we decrease the cost? When we were all growing up as students, taking money from home, staying away from home. So I am from Calcutta, I came to Delhi. I had to decrease my living cost. So I stayed in a shared apartment with 4 of us sharing the cost of living, food, and everything. So I could survive back in 2000 on a monthly 3,000 rupees from my home in Delhi by shared accommodation. Then I started earning, got my fellowship. First thing which I changed into a single occupancy apartment, because I found all the reason to convince me that I should live in a single accommodation. That's the reality. But then if you trace it back to genomics, how it is applicable? We can decrease cost of genomics, in my opinion, if we discuss, debate, and decide on doing one experiment which will help you ask 5 questions, 6 questions. Then if you do 10,000 divided by 5, your per question cost is 2,000. That's one way. Other way is that we are 6 like-minded PIs over here. We have questions which we want to ask. Can we frame those 5 questions in such a way that we do only one experiment? To any funding agency, I don't need to give it in writing that all experiments were done in my lab. Yes, we did this experiment and we solved 5 problems. So one of the things which we do in the lab is try to generate a data which we can, which will help us to ask all the questions which I talked about before. So one of the data set which we use is take clinical data in lot, spend a lot of weeks, not months, because that much time I don't prefer, lot of weeks on the clinical data to identify which patients you will like to understand in detail. Once you have identified those patients, we do, say, a host transcriptome. Now, the same host transcriptome data will help us or helps us to ask all these questions. How? Say, for example, I'm looking at a SARS-CoV-2 patient or a dengue patient. Both are RNA viruses. We find a lot of those reads also in our RNA-seq data. So if your lot of pathogen genome-related questions can be answered in that. And we have both published this as an idea as well as published independent many articles on all of these domains saying that same RNA-seq data has been used for this. Because it is, of course, you can, very straight question we can ask is looking at protein coding genes. The same RNA-seq data also has non-coding information or long non-coding RNAs. So we all know the functional role of those in disease dynamics. So we use that for investigating LNC RNAs. Same, different, one gene, one protein is not valid. So one gene can have multiple transcripts. Those transcripts may have different disease role. Many studies have shown it. So one of the study also which we do is to look into alternative splicing, that from can the same RNA-seq data be analyzed in a different way. Rather than looking at the gene, I also look at the different transcripts making that gene. So looking at the role of alternative splicing. And last but not the least is looking into role of non-human reads to understand co-infection or transcriptionally active microbes. So these are not the only 5, these are the 5 which we do in the lab because we can do only a certain number in the lab. But there are other questions which can also be framed and asked from the same RNA-seq data. So my one RNA-seq, which is 25,000 per sample for me, if I divide it by 5, it is 5,000. So I can say that, well, I can pitch to a funder that I need 25,000 to do this, but actually my cost is 5 because I'm going to pitch these 5 different studies to 5 different funders. So that way I can do multiple studies within that funding frame. So what I said, just a proof of concept of that same data has been used to investigate the whole dynamics of SARS-CoV-2. One of the work which shows that how SARS-CoV-2 lineage, SARS-CoV-2 between pre-VOC, Delta, Omicron and all varied in terms of single cell, LNC RNA role, and transcriptionally active microbes or co-infections. So this all from one single cell, from one RNA-seq data. Only difference is this is single cells. Of course, this is coming from single cell transcriptome, but the same, this is from the RNA-seq, this is from single cell. So if I do single cell transcriptome, again, using the same data, I ask all these 5 questions as 5 independent PhD problems in the lab. Yes.
What is the criteria to select that single cell? So which decides everything?
So
sometimes you don't know where exactly the population is high or in which phase that pathogen is there inside. So if you make some error in the selection, so that gives you wrong idea to continue.
Very right, very right. So what we do in this single cell work, at what we are doing in the lab is, we are not selecting the cells a priori to look at. So the question which we asked in the lab was that if there is a pool of patients who have different disease severity due to SARS-CoV-2 infection, mild, moderate, severe mortality, what is the role of cellular heterogeneity, or to be more specific, immune cells heterogeneity in deciding or underlying that disease severity. So we are not selecting particular immune cells to start off the experiment. What we are doing here is that how many cells would be sufficient to capture, because if I take 5 ml of blood and isolate PBMC, we get quite a few cells. Now, whether I should sequence 1,000 cells from an individual or 10,000 cells from an individual or 20,000 cells from an individual, that is a question which as a researcher you have to decide for yourself. If you ask company, they will say 20,000 cells. That's what they told us. Initially, of course, as a good child, we'll listen to what they told. So we initial first experiment, what we did was that we did 5 samples with 20,000, 5 same samples with 6,000, 6,000 and then 10,000 cells. And we say, okay, let us see. And then we generated the reads for it, and we found that 6,000 samples, 6,000 cells per individual is good enough to capture all the diversity of immune cells present in that context. How? We analyzed data for different cell numbers and we found that you are not increasing the discovery of more immune cells. Of course, you are adding up the numbers to those discoveries. So now what we do is that for each individual, we do 6,000 cells per individual, and then that is sufficient enough to capture the diversity and the dynamics of those. There are in-depth studies. If you are happy to listen, I have a lot of slides. I can speak till afternoon. So those, you will find some answers in those slides as well. So this is something what captures whatever we did during SARS-CoV-2 in a way, if I have to condense everything together. This is what Uma was saying, that when the first alpha variant came, we got a call from PSA office that we need to do sequencing at airport at 10 in the night. And I said, yes, we will do. Then, and we had to set it next day because everyone was supposed to come for inauguration as well. In India, we inaugurate everything, even in the middle of pandemic. So everybody was there to inaugurate, including international media, CNN and whatnot. They were there for capturing it as well. Very interesting. So I said, yes. So I said, okay, what is there we will get at the airport? They said, we will get this. This is literally a container. I said, fine. So people who have done experiments would realize that when you make sequencing library, you need one of the machine with centrifuge, which does take a lot of power. When we asked for what is available power to us, we found that centrifuges will not work. Okay, we said, okay, what to do? Because next day, 8 a.m., there is inauguration going to happen. So we thought, okay, what can replace those centrifuge? And again, this is sharing for you to think that, okay, we can innovate. So we thought, okay, what does centrifuge do? It literally helps to mix and then settle down. So we thought, okay, what else can replace it? So we went back to a vortexer and a spinner. But we have not used it before. I mean, we can't do it for international travelers and then leave a room for confusion matrix and saying, oh, this may be this and that. Because at that point of time, government was taking quarantine decisions based on the feedback which we're giving or the results which we're giving. So we had to test it overnight in the lab that whether your vortexer and spinner combination can replace the centrifuge. So we took 20 odd samples from the freezer, did library prep. Nanopores took 4 or 5 hours to make library prep, like 2 p.m. Around 2 a.m. we set up the sequencing. Because these were less number of samples, we ran it for 3-4 hours. We got decent number of reads, analyzed the data in quick time, and then got the answer that, okay, it is working. So in the morning, nobody knows that, few people know that stories that we had to work overnight because we had to replace the centrifuge with this combination of. Then when we went to the airport and set up the sequencing, we realized that bandwidth of data transfer was very bad because we needed to transfer the data to our servers to do the analysis. We talked to the airport authority. They said, no, we can't help because due to security issues, we can't open it. We said, what to do? Then again, MK1Cs, people who know nanopore different variants, you have MK1B, you have MK1C, you have gridiron, you have P2 solo, and you have promethion broadly. So we said, okay, luckily we had MK1C, which has onboard 1 terabyte space as well. So we were literally working in a way where sequencing was happening during the daytime because the protocol was that we will get all RT-PCR positive samples of international travelers at 6 in the evening at the airport. We'll do library prep overnight at the airport, put sequencing like 6 or 7 in the morning. Sequencing will happen till 4 or 5 in the evening, and that MK1C will come back to the lab. And another MK1C will go to the place for doing sequencing. So that way, we worked for a month and a half, where 2 MK1Cs were in circulation, because we didn't have the internet bandwidth to transfer the data. But what it taught us in the real time is that there is a solution to all the problems. And these are creditable solutions because our analysis was taken into account to decide if alpha variant, compulsory quarantine. If non-alpha, you can go for home quarantine at that point of time. This was in January 2021. Because it worked for that time, when Omicron came, because it originated outside India, we were again asked to do it. We said, okay, we were enthusiastic. But we didn't take into account that this time it was, because during alpha, because it originated from UK at that point of time, so the passengers only from UK flights were tested. Whereas during Omicron, it was expanded to all international travelers. So the load of individuals for which we had to do sequencing was manifold. But okay, we can't complain. So we did what we did. But so this was the lab which was functional again during the time of Omicron, and we had to share the data. So we were able to revert back to government in 30 hours time with respect to alpha, non-alpha during that wave, and Omicron, non-Omicron during the Omicron time. So that was one satisfying feeling, that you don't know the people for whom you are doing those work, but then your work is helping someone to go back home on home quarantine or had to stay back in the hospital, as that was the mandate at that point of time. So some of the key areas which we worked on is looking at international travelers, detection of different SARS-CoV-2 variants, because when we started in March 2020, we didn't knew that the journey will be so long, because we thought, okay, we have achieved everything. But then every 3-4 months, we had a new challenge or a new milestone to deal with. And that was a lifelong learning. And I say it with responsibility that people who work during COVID times have enriched themselves as a researcher or as a scientist for life to come, because that experience you won't get whatever simulation which we do in the lab. And we were literally working defined 24/7, because that's what we didn't have a choice. I had to sleep away from my wife for 2 years because I used to work in the lab. So I had to sleep outside in the outside room for those 2 years. And I was not allowed inside because my son was like 4 years old or so. So those were interesting 2 years. This is now this one. This is now used as a container. It has gone from there. So different.
Did you think of using a car battery to run a centrifuge? Because sometimes in the field we do that.
No.
Yeah, because that's also an option.
Possibly we could, but at that point, as I said, the time was like 10 p.m. And I said yes already, and then we realized that we didn't account for this. So all trial and testing happened overnight.
Oh, okay.
And the second point was that centrifuge which we had was quite bulky for anyone like me to pick it up. Yes. Yeah, we all discovered. There's a video, if we have time, I can play it at the last. It was very interesting, but we can laugh at it now.
So this is beyond the technical complications. So in your opinion, when you implemented sequencing, so let's say UK reported Alpha, South Africa reported Omicron, was it already too late to start sequencing? Or do you think you could have caught it and stopped it from coming in?
When we did this sequencing, it was already known. So it was like in the circulation for at least 15-20 days. So it was like part of India's response to track the dissemination into the community at that. So specific example to that is that when it was detected in India, December 2021, we had BA.1. And then we were doing daily sequencing for it. It took a month's time to evolve into BA.2 and then spread in the community as such. So initially we were having, like if you sequenced 50 samples during the day, your very high number or percentage were BA.1. But once it landed in India, because we have the advantage of quite a high denominator, so the virus got happy with finding so many new places to test itself. So it was important for government strategy to track how fast it is penetrating or expanding in the population as well. So it was not in the reporting per se, but it was like tracking the transmission in India.
So in wartime, money flows in, resources flow in, it's all good, right? But in peacetime, do you think there is a space for NGS to actually surveil for pathogens and locked down borders before pathogens make it into your country or our country? Whatever.
Short answer is yes. Long answer, as Uma said, that we are trying to get something to do in this domain for last 3 years. That's sad part of it. Good part is that we are still doing it for some pathogens which we can manage with the funding what we have. So for dengue, for last 3 years, we are doing very active surveillance to see whether what's the new mutations or whether there are new mutations in the dengue genome, whether any of them are associated with disease severity, how it is changing year by year. So we just communicated the findings of 2022. We have now done few thousand sequencing for 2023 and doing it with 2024. So we are doing what we can do, but of course answer is yes.
Point, this is probably a whole discussion, but this idea of taking things either to the airport or into the field or something which is very interesting and intriguing. And there is this, it's a Twitter handle, probably it's a company called Bento Lab, which I follow, and they make a lot of things. Of course, they're crazy expensive because it's somewhere in Europe or something. But I'm just thinking, because there are so many young students here, early career researchers, another thing to really nice opportunity to think about is to kind of miniaturize equipment, miniaturize things. How do we take it into the field? You had this kind of, was it a porta cabin or what would you call it?
This was like
a container.
Container. It's basically a container which is fabricated with some switches.
But there are some other people who are thinking about, kind of mobile vans where you can actually do this on the go. So it would be really interesting to do some of these experiments in India as well. Part of the make in India kind of, we can make it make in India.
Yeah, and these are very interesting things. Now when you think like, because when you change from centrifuge to vortexer and spinner, what is that which you change? And you have to change from plate to PCR tubes. So these are also things which goes hand to hand, that you can't then use your plates. Now your plates are non-functional. So those are things, then you need, then if you have those, then you need caps and all. So those were all things which we discovered that night. If it would not have worked, I don't know what would have happened, but anyways, it worked. So different aspects of when you look at pathogen from different perspective, you can either look at mutational dynamics. So I will cover this off in slightly one slider going forward. Okay, I'll not touch this, already talked about it. Yes, I'll talk from here. Okay. So I'll just go through some of the journey or the milestone during the SARS-CoV-2 times, as probably when I was thinking of how do I put my slides together, probably when we talk about the evolution of pathogens, evolution of labs are also quite important. That how does a lab evolves? Because you have to also communicate and convince your colleagues, your students that you are not doing routine surveillance. You are also doing something which is a hardcore research as well, because when a PhD student joins, after JRF, all PhD students want to solve the problem of the world. They are not at all happy doing genomic surveillance or RT-PCR or anything like that. So you are convinced, fine, but you have to convince your students because they are hard to, because they come with their own fellowship. You are not paying them as well. So we had also that luxury or in a way enjoyment to convince the students that what we are doing is a hardcore research as well. Why? Because you will need fund to do work. But once you do that work, you are at your luxury to use that data to the craziest imagination which you can think of. Nobody is going to control that imagination. That's your choice how much you can think of. So this is the first 125 SARS-CoV-2 genomes which we sequenced in IGIB in the month of April 2020. And these were the initial pan-India, we called it pan-India at that point of time, 125 genomes, because it was from more than one state. And we found that, okay, it has got this lineage, it has this distribution, and we thought, okay, very nice, we have done a decent job. And this traveled through many journals before it landed up in welcome open research. But once we were kind of feeling satisfied that we have done something good, we were soon given a reality check by initial murmurs and then realization that people who were earlier infected were also getting reinfected by SARS-CoV-2. So the question was that, is it like a reinfection or is it like a residual infection in those individuals for reasons we don't know? And this is all 3 years back, 3.5 years back, if you wind, there was not much information in that domain. So genomics gives you that toolkit to differentiate between two genomes by knowing that what are the mutations or the genome signatures which are common and different, and whether it is different enough to be called two different infections. So we took some, when we were encountering this, we found some reinfection cases. We took paired samples from those individuals, which was a rarity because normally after testing, it was not routine that you will store the samples because the load of the samples was quite high. So it was like routine clearance as well. So whatever we could find off, we could sequence few paired samples to show that yes, it was actual case of reinfection. And if it was not for Lancet, a paper coming in, this paper spent a month and a half in Lancet, and we were hopeful that it will be published in Lancet. But then at 10 a.m., similar paper came from a group in US, and 11 a.m. we got a rejection from Lancet. So that affirmed my conviction that I don't cite other papers. Reason being that we don't do bad science, and it's my strong conviction that we don't do bad science. It's just that we live in, or I live in Delhi, but all good science doesn't happen in San Diego. So good science also happens in Delhi, Bangalore, Hyderabad. So, and I wrote it back to the journal that this is so funny to note that your paper comes at 10 a.m. and we receive rejection at 11. After this was under review for a month and a half. And we were writing to them many times that given the sensitive nature of the work, I think you should make a decision fast. And there is not much things to make take so much of time because this is only 4 pair of samples. You just need to, but anyways, that's science politics at work. So whenever anyways, I'm a bit bitter on that. I'm trying to avoid using other term, but I'm bitter on it. The reason being that sitting on the other side, global north, you don't know better science. At least in genomics, few institutes in India do world-class science. And SARS-CoV-2 was a great normalizer that we showed what genomics-based science can happen in India when given a time. Anyways. So then as we moved along, the next journey, we thought now we have, answer to everything because vaccination has started. But again, we were proven, our happiness was proven short. We had vaccination breakthroughs. We were asked in a way that, okay, can we confirm that these are vaccination breakthroughs or these are like something else? Again, we took samples from individuals and tried to see whether there is any difference in terms of the SARS-CoV-2 genome signatures between vaccination breakthrough infections and unvaccinated yet infected individuals. And we found not much difference between the two, at least in the study what we did, except for a couple of mutations. But then as we went along and did some other work, that was not something significant which we could build on in future. Then as we moved along, we were now 2 years down the line. So we had the privilege or curse, whatever, to have few thousand samples sequenced in the lab across different time frame of SARS-CoV-2 footprint in India, from pre-variant of concern era to variant of concern alpha, delta, omicron. So we decided to see that, okay, can we use SARS-CoV-2 genome information and try to see that why some variants led to waves and not the others. So one of the way in which we looked at is try to look at the genome mutations and see whether they are, how loosely or how closely they want to be together. In other words, how good we as a friends would like to be. So if I am very friendly with you, then I will always go along with you. So even if in the trying situations, I will not like to leave you or go without you. So in that case, if there is a condition put that, okay, you can't enter here, two people can't enter here, then I will say that, okay, I will not go there. If you translate it back to pathogens, if there are certain genome backbone which is stringent, that, okay, I will not allow changes here, or then those pathogens may have a challenge with respect to surviving or increasing their number in individuals. So what we did, one of the approach we did, we tried to do linkage disequilibrium to see that which are the mutations, or are there mutations which are happily going together. To crystallize this, what we see here is we compared between, say, pre-Omicron, community samples and international travelers. And what we see here is that there is, in the pre-Omicron era, there are a set of mutations which are always together. So this darker the red means that they are always together. They don't want to change. Whereas when you look at the international travelers, you find that linkage to be heavily compromised. So there are not strong affection between mutations to always go together. So that's why possibly these are community transmission, that you start seeing these strong linkages breaking up. If you translate it back into a real life scenario, what it may mean like? It means that possibly the virus had more flexibility in terms of its genome architecture to migrate or to move from one individual to another, and that's possibly what drove some of the transmissions which happened internationally as well as in the community. We did a similar study for Alpha, Delta, and Omicron as well, and we found that the LD was strongest in Alpha, and then it was breaking down as we move from Alpha to Delta to Omicron. Then as we moved along, we tried to look at that, okay, there are a subset of individuals who don't survive, and there are individuals who survive. So we wanted to look at whether there are anything in the SARS-CoV-2 genomes present in those individuals which can give us some idea that why some individuals survive and some do not. So we looked again, basic analysis to identify mutations, then do group level analysis to look at it. At that point of time, we identified one mutation called S194L, which was present in the spike region, which was present in the patients who didn't survive compared to the patients who survived. Now, what may be the possible role of those based on the protein modeling work which we showed? It highlighted that probably it provided better opportunity for the virus to increase its number in the patients who didn't survive. Then question?
I mean, that mutation was seen in the spike region or in the polymerase region?
Spike.
Spike. Then how? Okay. Fine.
It was probably affecting the spike ACE2 binding for it to.
And I mean, a follow-up question. Did you look it in the perspective of host? Probably, some of the MHCs or any other immunological components were, affecting the mortality or.
Yes. I'm not sure whether I will have time to speak on it, but for the protein coding part of the work, from the same subset of these patients, we did the host study as well, and not for the same numbers because these are extremely high numbers. For RNA-seq, I can't do thousands of RNA-seq, but we did thousands of SARS-CoV-2 genomes. Then based on clinical data and the parameters of different severity, we identified subset of representative individuals. So for a host, we did around 250 RNA-seq for different severity to understand those questions. But yes, we did. So again, going forward, and these were a lot of things which were like having ear to the ground and trying to see whether we find evidence of that in the lab. So we were at some point during Delta's craziness, which we saw, there was literally a rush for looking for remdesivir to do something which may help. And we thought that, okay, is it something which is in the pathogen's genome which may be holding any clue that whether this patient may benefit or may not benefit. And we were not talking about the efficacy of remdesivir. We were just talking about, as a pathogen genomics, I have a bouquet of pathogen genomes, whether based on the mutations in those genomes with different batch to it, am I able to understand anything in that regard? So we did two-part study of it. One, we compared the patients who were infected by Delta variant and compared between individuals who survived and who did not survive. We were not expecting it, but what we found very surprising to our understanding is that when we looked for the binding region of remdesivir vis-a-vis the SARS-CoV-2 genome, we found that in the area where, in the patients who didn't survive compared to the, no, there is something wrong here. No, this is something wrong. But what we did find was that in the patients who survived, the binding domain or the binding of remdesivir was better in those individuals because of the mutation background it had, compared to the individuals who didn't survive. In those individuals, because of the background mutation which it had in that binding area, the binding of remdesivir was very weak, possibly. And again, we didn't go back and look at specific patients who did in that case, but then we thought that possibly it may be that whatever may be the efficacy of remdesivir was supposed to be in a subset of patients, it didn't have even those, because SARS-CoV-2 genomes in those individuals had specific mutations in those domains which didn't allow it to bind with better efficacy compared to the ones who recovered. And this was true for both patients of Delta recovery and mortality and Omicron recovery and mortality. And these were compared across, say, a gap of 6-7 months between these two waves which hit India. Yes. So this is something is like, this is Delta recovered patients compared with Delta mortality patients. So this is remdesivir binding protein simulation work to see where remdesivir binds with SARS-CoV-2, and we find this at better binding happening compared to the individuals who didn't survive. And once we became more wiser, we had all sort of information. For last 3 years, we tried to use some bit of machine learning or AI, because I was getting bit saturated to hear about AI. So I thought that, well, is it artificial AI better or actual AI? Actual intelligence is better or artificial intelligence is better? So we try to do a little bit more to educate ourselves and convince ourselves which is better. And I convinced myself that actual intelligence is better, because we decide or we define or we make these AIs. So I have this whole debate always. So this work was more to educate ourselves, that are people trying to scare me by saying that we have done AI, we have done LLM, we have done this. But if you break it down to actuality, what you are doing is that in one bucket you are having a sequence, then you are trying to find a pattern over there, and then you have trained me to say that this looks like a goat. And then you are putting me in a different area and putting maybe a different breed of goat and saying that, okay, can you identify it as a goat? So fine, I'm able to identify, but because first you trained me to see goat. But then it is important for that. So this was more for us and the students to use the data and convince ourselves that in science, what is also important, that whether we go for the terms, because they sound impressive, especially when you are in a PhD committee, students try to scare you by saying some high-tech terms, and especially if they know that you are not a bioinformatician, they will use some more jargons, kind of overpower you. But it is upon you to ask them to explain that, okay, tell me as if you are telling to your grandma who doesn't know anything. Break it down. So once you request anyone to break it down, you will realize that ultimately you are trying to find some pattern in a sequence. First you are trying to see that, and then you are given a new sequence to identify that, and that's what you try to see, that whether we can put that together. This is something what we did. So in last 3.5 years also, what we have done or tried to do is that we realized that when COVID came, we had few limitations in terms of where samples will go for sequencing. So there were places like NCBS Bangalore, CCMB Hyderabad, IGIB, NIBMG Kalyani. These are the few places where samples will go for sequencing. So for a country of 140 crore or 145 crore, whether 5 places are good enough for doing it on time, first point. Second, during COVID, we also realized that how do you ship those samples? Because I can complain later on that shipping conditions were not good, there was leakage, there may be possible contamination, technician didn't do a good job, didn't store at right temperature. Are we solution provider in some domain? Can we be solution provider in whatever we are supposed to be better at? So one of the concept which we have been pushing from that time is, can we set up micro labs for genomic surveillance in different parts of country, where samples can reach faster, detection can happen quicker, but it brings responsibility on us that if we claim that we have more experience on doing it, mark my words, I'm not using expertise because it's a very term which I don't use, because the day I become expert, I die. If I say that I'm experienced, then it's my responsibility to train new set of people to the best of my ability to use the sequencers, analyze the data, and make inferences. So we wrote to quite a few funding agencies with the idea that we want to set up micro labs in different parts of India with initial mentoring, hand-holding from our side, and then subsequently transfer over to them. So one of the initial partners or funding partner whom we convinced was AIDS Healthcare Foundation, AHF, to set up a micro lab at IIIT Hyderabad, which is a purely data science institute, no experiment, any bit of it. So we set up the first micro lab at IIIT Hyderabad, and it was launched with a lot of fanfare. Again, many people came for inauguration, including the Chief Minister's son at that point of time. People fondly call him KTR. So he was there. We have all photos on that. So we love, so that idea of taking, so then we took this idea to Wellcome Trust going forward, that one micro lab is not solution for India. So we said that, well, we want to set up few more. This time we were a bit ambitious to say that we want to set up 12 micro labs. Luckily, they agreed to the idea. They funded for it. But what we also did this time was saying that, well, if a new setup or a new place or a new organization or a new PI is setting up a lab, no fault of him or her, you also need to do quality check of the data being generated, both experimentally and analytically, to make sure that the work is done at the highest quality. When we say that Delhi is no less than San Diego, we have to stand responsible to that term. So we proposed another idea, that mega lab, that each of these 3 micro lab in certain part of India will be QC checked by a mega lab in the same region. So if there are labs around Hyderabad, the random 10% of samples from those places will go to CCMB for blind sequencing and analysis, followed by matching that whether everything's falling in line. So then they gave some funding. That's a project which is ending now. And these ideas were, our third organization also trusted us. We went to find Foundation for Innovative Diagnostics. So they funded for 3 micro labs. So during COVID, we set up these 16 micro labs in different parts of India with mentorship and now transferring. So in September, I'm going to formally transfer that IIIT micro lab to the micro lab to IIIT Hyderabad for them to run it for their other programs. And that was acknowledged with WHO as well, that this is something which will not only lead to capacity building, it will also be a step towards pandemic preparedness, the point which you said, that it has to be long-term investment. We cannot react because yesterday there was rain and I can't go and buy a raincoat. If I am even minutely intelligent, I have to see whether it's a pattern here. If it is one day, I can still manage either work from home or using umbrella. But if it is a place where it rains every now and then probably I can go and buy a raincoat. So if we are not saturated, you please ask questions. And if you're still interested, I will speak for more I have only covered 1/3, 2/3 is left. Oh, what's the time? The advantage of not seeing the clock on the side.
One query. Does your institute, kind of creates any workshops where we can come and learn this NGS or?
Dekho, don't worry about institute. I will leave my contact details. Write to me. I am a firm believer of doing at your end. If you want to, get everyone to join you, it takes longer. So if you are interested or 3 of you are interested, I won't wait for my institute to do that. I do it quite routinely in the lab, for which I don't need any permission or don't need any money. I have funding support to take care for this. So it's a more of a like based on interest. If you are interested, we can arrange a time. You can spend 3 days, 4 days at IGIB, see in real time. I won't organize a workshop in that sense, but because these are routine activities in the lab, you can work with the students and learn in real time that, okay, this is what is happening. So there are students who, even if you go today, they will do this, somebody is doing this, somebody is doing. So shadow them, learn them. Analysis is happening in the real time. Which domain you want to, you have a strength and you want to devote more time, you want to know the both. This happens in real time, but you are happy to reach me at any point and it can be done. So don't worry about the institute part of it.
Thank you so much.
And I think in a way, of course, these kind of big workshops also good, but actually embedding in a lab is a really great way to learn stuff.
The reason being that last year we organized an Indo-Vietnam workshop. And because it was an international workshop, so there is so much of permissions to be taken. Because these are international faculties, so we had to take all those clearance from MEA to headquarter. So this year again, we are organizing an Indo-Vietnam workshop for 21 faculties from Vietnam. But we have started now. So we know that the permissions will take 3 months. For the academic part of it, for the workshop part of it, it's a routine lab work, so that's not much, it will take time. But so that's why where it is manageable, because for them, I can't do it in the lab because those are international travelers, so it has to come through clearance of MEA. Even if I want, I can't say yes. But for us in India, I can avoid that route unless there is a really good number to organize it in a formal way. And before I take that question, we are also, for example, trying to figure out, for example, we are setting up a center of excellence with BD to do twice a year single cell multiomics workshop, where the idea is that in partnership with us, they will take care of the logistics, because I get tired with logistics. So I need someone who can take care of the logistics. I'm happy to do what the scientific part of it requires. So we at the moment plan to do at least twice a year, single cell multiomics. BD, Beckett. BD. Beckman Coulter. No, Beckman Dickinson or something. That is for flow cytometry. Flow cytometry, synonymous with flow cytometry. They have newly ventured in single cell domain. BD Rhapsody, that's the platform which I use for the work. So when we did quite well with initial exposure to it, so now we are entering into a MOU to set up a center of excellence, at least for next 3 years, where they will take care of the logistics and reagents. Single cell work are costly. Each sample single cell transcriptome of say 6,000 cells per sample, our cost is 1.25 lakhs. And it doesn't include infrastructure, it doesn't include data infrastructure background, which your servers, GPUs, which are a very significant component. So it is a costly affair, but the kind of understanding you will get is immense as well. So that's where if you remember one of the slides, that how many questions you can ask from the same data. And before I wind up, I'll share one slide that how we did a work on investigating intracellular microbes from the single cell data. So that is still, as I speak, is the only publication in the world where we have shown that using single cell transcriptomics, you can also look at microbes present within specific immune cell type and potential role of those microbes in modulating those immune cell types, only from genomics perspective. And still that's the only paper in this domain. Of course, because publishing from India, we published in iScience, but it could have landed in Cell if it was from Harvard, but no worries.
So I have two questions. Like I saw you are doing some genomics in now shifted to dengue and
Lot of genomics.
Tuberculosis. So
Tuberculosis is still baby child.
Yes. So already you have established a system which was working for COVID nicely. So why you have not picked up like something similar like flu or surveillance, which is
Good point. So on the similarity index, I can defend dengue because it is also a single-strand RNA virus. The second question, which we debate and discuss in the lab before deciding is, if you remember the first slide, that what work which we are doing is relevant for the time and the patient in the hospital. If I talk about Delhi at this point of time, and which is true for possibly Bangalore and Hyderabad, other places as well, that dengue is something which is on the calendar on very high number starting from July to November. If you go by the numbers of Government of India, I have more than that number of samples from one hospital in my lab. So it's a comment on what is the penetrance of, say, dengue in terms of its incidence in India, and what may be the implications of that. Why it is important? Because one, you feel you need to have incentive, and this is more like I speak from my own or lab perspective, that some incentive to do work as well. That I may decide to solve a problem of ribosome structure. Very good. But as a PI, I don't feel incentivized. I feel incentivized is some of the work which I'm doing is helping some patient whom I don't know. So some of the work, so going by, say, end of the year, for example, we are in partnership with clinician planning to include 2 gene for the RT-PCR of dengue patients. At the moment as we speak, we don't do RT-PCR for dengue detection. We do NS1 or IgG and say that it's dengue positive. Whereas during SARS-CoV-2, we had a whole network or infrastructure for RT-PCR, but those RT-PCR machines are not being utilized. Can whatever work which we do in the lab can be translated to RT-PCR strategy, not only for dengue detection, but also for dengue severity by including genes from host and pathogen. So that's work which may not happen next week, but by end of the year, that's the plan which we intend to do. And that's why we picked up dengue as another disease to focus on. Other diseases, I actually received a message in the morning that can we work on other diseases? I said, well, it's a bit troublesome if you scratch too many surfaces and don't do anything deeper. Then the things which I don't like, I will not be liked for same things. That I'm trying to be an expert of everything. I probably could have worked deeper on SARS-CoV-2, but then at the moment I can't find clinical patients. And I do intend to work in that clinical domain area. If there were, and this is sounding nasty, but then if SARS-CoV-2 could have been like, say, dengue, for example, I would have loved to go deeper, more to understand many facets of it. But
what about influenza?
Yes, but then as I said, that if some more funders join us, we will be with a group of people, because we are at like 30 odd people in the lab, and all those areas are individual research problems as well. So you need that sort of a mandate as well. But that was our rationale to pick up dengue to concentrate deeper on it. MTB, we are working on a very specific aspect of MTB. Maybe I should clarify. We are trying to work on female genital tuberculosis. That's the area in terms of, so we are not working on classical TB in that sense. So we are trying to work on understand the genomic dynamics of female genital tuberculosis. That's because through one of our clinical partners, we realized that around 30% of infertility in females, 30% have tuberculosis in the genital area. So we want to understand whether that is contributing in any way to infertility. Some of the rationales which we put together to convince AIDS Healthcare Foundation for funding was that in the pH of the residency of the bacteria, there is very different from the normal human body pH. So how is MTB adapting to that microenvironment as well, and is it contributing? So that's a project which just got initiated, not much insights, but hopefully we will learn something in that process.
Can I ask a question? So you showed one of your study where you were comparing the alpha, delta, and Omicron, looking at the linkage disequilibrium, right? And you were saying that the association between some of the mutations or the thing found in alpha was more. As the variant diverged, like Omicron, it became, the pattern was not so. And I think one of the comment you made was that that kind of tells what's going to cause the wave and what may not, right? So now going forward, like since Omicron came out, there were only Omicron divergence in so many shape and form. Yet some of the Omicrons have caused very larger wave than the others. If you were to do this analysis again, looking into the patients, how do you think that would pan out, that kind of analysis?
Very good question. And luckily for us, we have done that study, incidentally not in the deck of slides which was there. So when these were the times of recombinants coming in, and we wanted to understand that why recombinants at this point of time. One reason can be that analytical pipeline has started detecting this, which it was not capable of, or which it not detect before. So what we did first to answer that question to us, for all the 25,000 plus genomes which we have sequenced in the lab, we analyzed that data all over again with the latest pipeline to first tick off that whether it is anything to do with the analytical sensitivity of detection. And the answer was no. It did not detect any recombinant in the previous generations. So that was satisfying to a way, so that we said, okay, why it is happening now? So in the different recombinant lineages again, because there are so many XBB dot this as well. So what we found in that study as well, that the logic which we found here, that the breakdown of your LD was happening as you studied this. Similar thing was happening when you look at the recombinants. But if you compare it with the base of alpha, you won't be able to capture it. But if you compare it with the base of Omicron, in different subsequent lineages of Omicron, you will find that your LD is breaking down further in terms of it. That was observation. But what is inference of it, or what was our logic towards it? We thought that why did genomes started recombination at this point of time? Our idea was, again, it is subject to a lot of other alternative opinions, is that SARS-CoV-2 has been trying quite a few ways to survive in different situations with the bigger disruption through vaccination and other stuff. So at some point it decided that I need to make bigger changes now. Probably single base changes is not going to be sufficient for me. Probably I need to make more fragment level changes in that direction. That's possibly why we detected recombinations as well. But the observation that your LD is breaking down since the first ones stands true even for these ones. And that we published, I think, 6 months or odd before, from a different perspective actually. If you see the title of the paper, we asked that when these lineages are evolving from compared to wild type, alpha, delta, omicron, alpha had, for example, 18 odd mutations. Delta had 32 mutations. Omicron had 52 mutations on average, I'm saying, compared to wild type. These mutations, if you translate into ATGC, we wanted to understand that whether it is leading to disequilibrium of ATGC proportion as well. So from that perspective, we asked that question, that whether these different lineages are leading to this disequilibrium, and which gets more explicit in recombinants because you are making bigger chunk changes in that. So that we did find that your ATGC dynamics is changing, and it is not random in nature. So there is a directionality in that ATGC proportion changing, and which is getting reflected or getting association with the severity of the disease as well.
This is so strong. I mean, I think so as soon as a variant of concern or interest or under consideration, whatever is discovered, I think if this analysis helps, then it can be used, right? Because, so for example, the January 2022 first Omicron wave was big, but XBB wave was even larger than that. So comparing that with that, I think would be very interesting. And then JN1 came, which was not a very strong wave, basically. So what I'm saying is like predicting that how just based on this kind of thing, I think would be very useful, based on clinical data, of course.
Yes,
So yeah.
yes. I agree with that.
Sir, second question. Sir, I am from vaccine industry background, and we are always like continuously trying to improve existing vaccines, and we are developing new vaccines also. And as you said that we are doing good in our science, and like we are having expertise, and regarding that we want to, so regarding that we want to collaborate with the like those who are in the government, and those who are having expertise in that domain, but it is difficult for us to get connected with the team members, and after getting connected again it will go to the director, and then our questions will die in between, like somewhere. So how we can solve these problems?
One-to-one. One-to-one, why I say this?
I think that's, yeah.
So for example, say you are interested to, work on this domain, for example. It's like working between one lab and say your lab or your company is a better idea. That's what I did with BD. That instead of making a CSIR BD, it's a too humongous work to do. Let's do it at the lab level. So we are setting up a COE with my lab and BD as a partner. It is easier for me to drive it that way. If you are interested, for example, it's more like one-to-one. We'll build very specific, and there are provisions within government institute to do it. For example, we have a body within the institute called BDMG, Business Development and stuff. So that's a body within. So for any proposals in this domain which may have a potential wealth, money related to it, we submit a proposal through BDMG, and we can do that project as an industry academic partnership. So there are provisions to do it. It does sound scary, but now as I have lost more hairs now, it is doable. That's my sense of the matter is that it's doable.
Great.
Just one slide to make justice to the funders who have funded, because without them, I'm nothing. And what you see here is you will not find much of government funders, because they take a little bit of energy from you rather than give you. So I have been more happy to convince the international ones, and luckily they have been able to trust what we proposed. So a lot of work which we did is through all these funding partners. Couple of them have supported us second time as well. So that reflects that we didn't do bad job at the first time. And these are group of 30 odd people. We spend together, go sometime on lab retreat, but they do complain that I again go and talk about work in lab retreat. So that's a complaint which I have not been able to remove at the moment. So hopefully we are planning to.
You should visit us in the field sometime.
That's why I proposed that Kolar stuff when I saw it. I said, well, that's a good place to go.
Yeah.
But this July again, we are probably going to go on a lab retreat. So with that, I'll stop.
Thank you so much. Thank you so much.